Hello everyone, this is Dr. Young, and in this first video with proteins and amino acids, we're going to talk about amino acids. We're going to focus on kind of like what they are, what their structure's like, how we can draw them, and then in the next video we'll talk more about how do we classify amino acids, how do we make dipeptides, polypeptides, and then in the last video we'll talk about protein structure. So what is the difference between the primary, secondary, and tertiary structure of a protein? So in this first one, I just want to introduce proteins to you and start talking about what amino acids are. So for most people, right, when you hear protein, you think of probably like diets and food and stuff like that, right? The grams of protein are on your nutritional labels. There's a lot of high protein diets out there right now, like an Atkins diet, or the current fad is the keto diet, right, which has relatively high amounts of protein. But proteins are really, really important because you are mostly protein, right? So I'll just put a little note here that after water, you are primarily made of protein. You're a protein machine. You're primarily made from proteins. Given that body fat may vary and everything, but as far as functionally goes, you're mostly proteins. And those proteins are super, super important. They do so many different things. And I've listed here just some of the many functions, right? Oftentimes when we think of protein, like in us, we think of like muscle tissues and stuff like that. That'd be an example of contractile proteins, where we have smooth muscle, cardiac muscle, skeletal muscle, right? So we have muscle tissues made out of things like actin, myosin, titin, proteins like those. We have some that act as hormones, right? So these would be chemical signals. I'll make a little note of that, right? So when we're talking about hormones, we're talking about anything. A hormone is any molecule that is a chemical messenger. So we're talking about chemical messengers here. So chemical messenger. And so some hormones are proteins, some hormones are steroids. We'll talk about steroids later. But any hormone just sends a signal, basically. So I gave two examples of proteins here, right? So insulin, that's the one that signals for your cells to open up and allow glucose to come in. Oxytocin, sometimes called the love hormone, or it's that affection, that bonding hormone. That oxytocin's sole function is to just behave as a signal. Same thing with insulin. They don't do anything. They don't change anything. They don't make sugar. They don't make love. They just signal for the body to do things. They start cascades. We have transport things. You guys have probably all heard of hemoglobin, right? It transports gases around in the blood. You may have heard of HDL and LDL. Those stand for high-density lipoprotein and low-density lipoprotein. Those are used for transporting lipids around, specifically cholesterol. So you might have heard about those lipoproteins. They're proteins. They just move stuff around. And there's other examples. In your biology classes, you'll talk about sodium potassium pumps and GLUT1 and all of these proteins on membranes that help transport things in and out. Enzymes, you may have heard of things like lactase, maybe even amylase or something like that, right? Enzymes just help reactions go faster. They help lower the activation energy of something. They just act as catalysts. I'll make a little note of that, right? We're talking about just catalysts. If you've heard of catalysts before, right, they just speed up reactions. So you have tons of different enzymes that help speed up reactions in your body, in your cells. And then lastly, there are some structural proteins also. So these are things where their sole existence is just to give structure to something, right? So like the keratin in your hair and your fingernails, like collagen in your skin, or elastin to help keep things kind of in place so that your liquidy cells just don't like droop and blob all over the place, right? These just give structure to your body. And these are just some of the functions and just some of the proteins. There's so many. Proteins, in my opinion, are hands down the most important biomolecule, which is why, right, DNA codes for proteins. It tells your cells how to make proteins. There are so many different types. So let's take a little closer look at what proteins are made of. So if you take a look at proteins, right, so we have our classic macromolecule subunit distinction here. So proteins, right, proteins would be your macromolecule because they are made up of many individual amino acids, right? So then your amino acids would be your subunits. Your amino acids would be your subunits. And you may see proteins are sometimes called polypeptides, right? Poly meaning many, right? So you might see poly here meaning many. So many. And then the word peptide, peptide just means, it's another word for an amino acid. So by polypeptide, we mean many amino acids. And these amino acids, right, are combined, just like we saw with the nucleic acids or all those examples with condensation and hydrolysis. The idea is that you have all these individual amino acids and then they get condensed to form linkages, so they start to become one larger macromolecule, so one big macromolecule. And proteins can be dozens, hundreds, thousands of amino acids long. And so I have down here an example of a protein, and the idea is that if you took a closer look at this, what you would expect to see here, if I zoomed way in, and we'll look at some structures in a second, is that this is a representation trying to show all the amino acids. And what you're not seeing is that it's like there's one amino acid connected to another, and another, and another, and another, and another, and that this whole long chain is just a series of amino acids that are connected to each other in this really, really complicated form to give you this protein. I'll stop there, but you can imagine the whole thing would just be hundreds of amino acids connected end to end, one long chain, one long macromolecule. And so in order to understand this protein a little bit better, which we will do in a couple of videos, we need to understand the amino acids a little bit better, because they're the ones who decide why we get, for example, these curlicue things, or why, for example, we might get things that these sheets that go back and forth like this. Why are some things on the inside of a protein, some things on the outside of a protein? So we need to learn a little bit more about what an amino acid is, what the different types are, how do we classify them in order to understand that a little bit better. So let's do that. So for your amino acids, I have a couple of examples here. I have for you, this is valine, one of those branched chain amino acids. This is serine. And I'll mention this again later, but for my students, I don't ask you to memorize the 22 structural amino acids. I'm just giving you names, so in case you've heard them somewhere, you know what I'm talking about here. And so our idea, right, is that our amino acids, they have 4 groups. They have 4 very specific groups around a central carbon. So I'll put around a central carbon. And this central carbon is sometimes called the alpha carbon. It's the carbon that's in the middle. So the idea is that I have some sort of carbon with 4 different things sticking off of it. And so for my 2 examples here, valine and serine, I'm talking about this carbon right here and this carbon right here. Those are both going to be my central carbon, or my alpha carbons. And so the first thing that you're going to see coming off of all of them is an amino group. And there's no particular order here, I'm just putting them down. But one thing that you're going to see, right, is an amino group. So your NH2. So I'll add an NH2 to this down here, and I will highlight over on my molecules that I have drawn. I will highlight, right, here's an NH2, there's my amino group, there's my amino group. So I do have an amino. Right, you have an amino group on your amino acid. That's why they're called amino acids. And then they're called acids, right, because they have the acidic functional group. They have the carboxylic acids. So the second thing you're going to see is a carboxylic acid, or a carboxyl. You're going to see a carboxyl group, so that's CO2H group, right? So you're always going to see a C with a double bond O and an OH. Here's your carboxyl, here's your carboxyl. So we have carboxyls also. So you can identify it. You'll notice that these are both coming off of the central carbon. They're not just somewhere randomly on a molecule. They're coming off of this central carbon, this alpha carbon. We will also see something that's kind of boring. You're just going to see a whole bunch of these everywhere, right? You're always going to see a hydrogen, which is a boring CH. So you're always going to have an H sticking off of these. So I'll add an H here and an H here. So I've got some hydrogens sticking off. I didn't draw them, right? This is a zigzag drawing, that skeletal drawing, line bond. So I didn't draw an H coming off that carbon, but there is that hydrogen there. So carbon has 4 bonds. And then the last thing is called the R group, or the residual group. So thing 4 is the residual group. The residual group, right, which we just label as an R, capital R. This is sometimes also called the side chain. And this, I put R, right, because it can be kind of anything. It can be just kind of whatever. And it's this R group, this residual group, that defines the amino acid, that tells you that one thing is serine and one thing is valine, right? Because if you look at my 2 amino acids that I drew right now, the things that I circled and highlighted are identical, right? I have a carbon with an amino, with a carboxyl, with an H on both of them. That's the same thing. But what's different is that valine has this V-looking thing, right, this CH with 2 CH3s on it, versus serine has something different, right? So that would be the R group, that side chain or that residue group for that amino acid. And again, that kind of tells you which amino acid you have. So amino acids all look very, very similar. They have amino groups and carboxylic acid groups, and then that side chain is what tells you what amino acid they are and describes their properties better and where you find them in proteins. So those are the 4 groups that I'm looking for on an amino acid. If it doesn't have those 4 groups, it's not an amino acid. If it doesn't have them around the same carbon, then it's not the kind of amino acid we're talking about. It's not an amino acid, which is not an alpha amino acid, which is the kind of common amino acid that we're talking about, right? So if I had, for example, a molecule that looked like this, and let's say we had something like this, we would not consider this a amino acid, right? Not a normal amino acid, not the one that we're talking about at this level, right? So not an amino acid. Yes, it has an amino group. Yes, it has a carboxyl group. Yes, it has a hydrogen somewhere. There's lots of hydrogens, but my problem is that I don't have a central carbon that all those things are coming off of, right? If I'm looking at the carboxyl, that's coming off of this carbon, but that carbon that I just drew in black is not the same carbon that the amino group is coming off, right? This amino group is coming off of this carbon. So I don't really have a central carbon, I have 2 different carbons. And so again, the idea is that you have to have an alpha carbon. There needs to be one that's in the center here, right? So that like this would be the alpha carbon in that one, this would be the alpha carbon in that one. But here, I don't know which one's alpha, I don't know which one's the centerpiece. They're just kind of spread out in the molecule. This is not an amino acid. Now, when we look at these amino acids, right, you're going to see them drawn in one of 2 ways. They're both fine, they're both right. It's just that because they look different, sometimes students freak out and they're like, oh God, is this an amino acid or not? What's going on? And so over here on the left, I drew kind of your classic version. I just kind of blew up serine again. So this is serine. And what you'll notice here, right, so on the left, I'll circle this in blue, right, I have my amino group. This is my amino group, my NH2. And amino groups are basic. They're basic. And you may or may not know what acids and bases are yet at this point, but basic just means that they want to accept an H+. They want to accept an H+ from something, from somewhere. And they want to pick that H+ up. And if you look at the carboxylic acid, right, just like its name suggests, if you look at the carboxylic acid, the carboxyl group, so carboxylic acid, right? The word is acid, which means that it wants to donate. It wants to donate an H+. It wants to donate an H+. It wants to give up an H+. And the idea is that this is the H that it wants to donate. This hydrogen in the carboxylic acid is the H that it wants to give up, it wants to donate. So it wants to get rid of that H+, and then this nitrogen over here actually wants to pick up that H+. It wants that H+, it wants to pick it up because it's basic. And so what you see is that serine can be drawn like I drew on the left, or it can be drawn like I drew on the right here. So you'll notice, right, that this nitrogen has an extra hydrogen. It picked up this hydrogen. But by picking up an H+, not only did it pick up a H, it also picked up that positive charge. So it became NH3+. And then likewise, that serine, the carboxylic acid on serine, over here on the right-hand side, there's no more H. This H that was right here is gone. And since it lost an H and a positive sign, right, if it loses a positive sign, it became negative. And so what we see is that these are 2 different ways to draw serine. Sometimes you'll see serine drawn like it is on the left. Sometimes you'll see serine drawn on the right. They're both okay. They're both correct. They're both fine. You're going to see them both different places. Now, technically, the one on the right is better than the one on the left. Technically, it's not better. It's more realistic. It represents it better. Really, it looks like the one on the right versus the left. There is a difference here. So if I look at these, right, on the left-hand side, I have what's called the non-ionized form, right, because there's no ion here, right, so this side is the non-ionized version, right, because I don't have any ions. It's not ionized, no ions. I don't have any charges. Versus the way that we draw it on the right is called the ionized version, the ionized form, or you might hear it called the Zwitter ion. Not zwitterion, it's the Zwitter ion. The Zwitter ion just means that you have a positive and a negative in the same molecule, like we do here, right? We have a positive on the N and a negative on the O. And so you can see serine drawn out like this too, which is also the same thing as saying that we have NH3 plus attached to a molecule that has this OH on it, whatever that R group is, and then O minus. So sorry, let me actually erase this so I can show you the condensed form. What you might also see is CO2 minus. Same thing. So all three of these drawings really are the same thing. It's just that the one on the left is the non-ionized, and the two on the right are considered the ionized forms, or the Zwitter-ion forms. Again, you'll see them both. Technically, the ones on the right are more correct, and in physiologically speaking, that's what you're more likely to see. I don't care which one you draw. Again, I'm just going to ask you, what are these? And I'm mentioning this mostly because when you look out, on the internet, when you look in a textbook, some people are going to use one form, some people are going to use the other form. It's the same thing. So for example, here's one I got off the internet. This is off of Wikipedia. This is not even in English. You'll notice that the names are all kind of funny. But the idea is that if you take a look at this, right, and I want to know, is this in the ionic form or the non-ionized form, right? I don't see any charges, right? This still has its H. This didn't pick up the H. None of these, right? I didn't move the H at all in this case. So I'd say in this case, we're talking about all of these are in the non-ionized form. Someone drew all of these in the non-ionized form, which is perfectly fine. It's totally fine. These are all the 20 structural amino acids. Whereas if you go here, right, what you'll notice is that if you take a close look at any of these, what I see is, oh look, this is NH3 plus and this is COO minus, right? This does not have the H anymore. This does have the H. The amino did pick up the H, so this would be an example of something that is drawn in the ionized form. So here, all of these 20 structural amino acids are drawn in the ionized form. And again, it's not that one is necessarily right or wrong. It's just people draw them different ways. It's the same thing. Don't freak out. So hopefully at the end of this video, right, you have a little better sense of what proteins do and how proteins are made up of amino acids. There are lots of different amino acids. There's 20 to 22 that show up in proteins. There's way more than that. I don't expect you to memorize these 20 amino acids, but what I do want you to be able to do is classify them. Which ones are acidic, which ones are basic, which ones are polar or non-polar, which can be phosphorylated, etc., which is what I'm going to talk about in the next video. So if you have any questions or comments, please go ahead and leave them down below. Study up and then watch our second video where we will, again, classify those amino acids and make small dipeptides, polypeptides with them. Happy studying.